Welcome, my name is Samantha Llewellyn and I am a researcher working on the Patrols project at Swansea University. Today I will give you an insight into the development of an advanced in vitro 3D liver model for nanomaterial testing. As a bit of an overview, the liver is the detoxification centre of the human body and the main site of secondary nanomaterial deposition following either primary route of exposure, so ingestion, inhalation, injection and dermal penetration. Yet less than 1% of the inhaled nanomaterial dose actually makes it to the liver. So the liver itself consists of two main lobes, both of which have 8 segments that embody thousands of liver lobules. These lobules are hexagonal structures constructed by repeating segments radiating out from the central vein. Each segment, as shown here, links to one another by the liver sinusoids and consists of the portal triad, so the bile duct, portal vein, and hepatic arteriole. Within the liver, there are two main cell populations, parenchymal and non-parenchymal cells. Approximately 80% of the liver volume is occupied by parenchymal cells, whilst the remaining non-parenchymal fraction is made up of endothelial cells, stellate cells, and Kupffer cells. These cells work together to maintain metabolic balance and detoxify any endogenous or exogenous toxins. Whilst it is known the liver can regenerate, even back to its full size from as little as 51% of the original mass, there is only so much damage that it can take before the progression towards permanent liver disease begins. So for example, this slide demonstrates the progression of liver disease from a healthy liver all the way through to liver cancer. So it begins with a healthy liver that is able to effectively perform its normal functions, helping digestion and breaking down harmful drugs and poisons, to a fibrotic liver, which arises as a result of damaged cells which promote continuous inflammation of the liver, leading to the accumulation of extracellular matrix and the formation of scar tissue, which often stiffens the liver and causes irregular blood flow. So with this restricted blood flow comes limited gaseous exchange and nutrients able to help keep the hepatocytes alive, and so excessive hepatic apoptosis and the deposition of scar tissue replaces normal, healthy tissues. Now, this often results in the blocking of further blood flow through the liver and prevents normal liver functionality, leading to the cirrhosis of the liver. Then the final stage of liver disease often ends with liver cancer, and this arises as a result of DNA mutations and alterations in proto-oncogenes, tumor suppressor genes, and growth factors, and all of which can lead to uncontrolled cellular proliferation and the formation of malignant tumors. So as you can see, whilst there are a number of different manifestations of liver disease, there is a natural progression with key events which can be targeted for the early detection and prediction of these adverse outcomes. As a result, there is a need for more physiologically relevant and reproducible test systems able to aid a high throughput nanomaterial hazard assessment and predict any potential adverse outcomes which may arise as a result of nanomaterial exposure. So with a vast variety of nanomaterials on the market, it is unsustainable to rely solely on animal-based testing methods. And so there's been a major research drive aligned with the 3R principles listed here to find and develop advanced in vitro liver models that have the potential to provide suitable in vivo alternative test systems for nanomaterial testing. So here are just a few examples of in vitro test systems that are currently on the market, which then leads us on nicely onto the development of our 3D in vitro liver model. Based on two principles, one being that the primary human cell types can exhibit high interdonor variability, and the second being that standard 2D cell cultures don't encapsulate the intricate multicellular interactions and metabolic activity observed in vivo as well as the 3D structures, we began to develop a 3D cell line-based liver model. The aim of this work was to advance an existing 3D in vitro liver model and enhance the physiological relevance, improve the capability with toxicity endpoint tests, and develop a 3D model able to remain viable for 14 days in culture. So this was to allow for both acute, prolonged, and repeated nanomaterial exposure regimes to be evaluated. So our model itself is established using a technique called the hanging drop method, which forms the 3D liver structures known as spheroids using gravity alone. So to help with the visualization of this process, I have a quick video to show you the technique we use for seeding the HepG2 cells into the hanging drops and to allow the spheroid formation process to begin. For creating the hanging drops, required to form the spheroids can be quite technical, but with a little practice, everyone can do it. So in short, we pipette drops of the Hep G2 cell suspension onto the lids of the 96 well plates. We then flip the lid and leave to hang for 3 days to allow the spheroids to form. So each spheroid consists of about 4,000 Hep G2 cells. And once the spheroid has formed into a compact, ball-like structure, we can then transfer the spheroid from the hanging drop position into the base of the plate, ready for nanomaterial exposure. And that is indicated here on the picture on the right, where you can just about see the little spheroids in the center of the wells. And they are highlighted there with the white arrow. Once this model was optimized, we fully characterized it over a 14-day culture period and published the method in the Jove journal earlier last year. This 3D liver spheroid model has been shown to remain over 70% viable following 10 days in culture, 14 days if you include seeding time, and retain its liver-like functionality through the increased production of albumin and urea all the way up until day 14, where it begins to tail off in parallel to the drop in cell viability as seen in A. This model is easily accessible, simple to construct, and quite adaptable, allowing us to add additional features, which I will come back to later on. Following nanomaterial exposure, the supernatant and spheroids can be harvested simultaneously or independently if necessary. As a result, this 3D liver model is compatible with a number of biochemical endpoints, from cytotoxicity and liver functionality to inflammatory markers, gene expression, and even DNA damage. One key feature of this proliferating 3D in vitro model is the ability to support fixed DNA damage assessment via the micronucleus assay. So with further advancement of these models to improve their predictivity, they could provide suitable alternatives to animal-based testing methods for high-throughput toxicological screening of nanomaterials. To assess the transferability and reproducibility of the model, an interlaboratory trial was recently undertaken. We found that the model was easily transferable with only a few minor teething issues which were easily resolved with further detail clarified in the SOP. So that in this part of the interlab trial, the model was transferred from the host lab, so lab 1, to an industrial CRO, BioReliance, lab 2, for genotoxicity assessment. So both labs assessed acute 24-hour exposure to a positive control chemical, benzoapyrene, across a similar dose range from 0 to 8 micromolar, as shown here in Table 1. For genotoxicity, the lowest observed effect level, at which the first significant induction of micronuclei was observed, was almost identical between the two labs, with a similar fold change in micronuclei seen. For the interlab trial done within Patrols itself, a longer-term 5-day exposure to barium sulfate nanomaterials was tested in both the host lab, Lab 1, Swansea, and the transfer lab, Lab 2, Heriot-Watt. The IL-8 pro-inflammatory response was assessed comparatively in this trial, following prolonged exposure to 0.2 and 2 micrograms per mil of barium sulfate nanomaterials. As is seen in this figure here, there was good concordance in the negative untreated control, with no significant difference between the IL-8 response observed in the Hep G2 spheroids from Heriot-Watt Uni and Swansea Uni following the 5-day exposure to barium sulfate. Both of these trials demonstrate the successful transfer of the protocol across laboratories with excellent reproducibility in hazard endpoints. This final slide just highlights the development process undertaken for enhancing the physiological relevance of the 3D liver models from the baseline Hep G2 model described in the previous slides to where we finished the Patrols project. Initial progression involved enhancing the model itself to include the addition of secondary cell types. In this case, it was the addition of liver-resident macrophages, known as Kupffer cells, as these are the main cells required for initiating an immune response to help clear the foreign material. To replicate human physiology, for every 4 Hep G2 hepatocytes, we added 1 Kupffer cell. This ratio worked really well as we saw great integration of the 2 cell types within the spheroid, as shown here in this confocal microscopy image. Next, we looked to adapt the environmental conditions by replicating the oxygen concentrations available in the liver within the human body. Atmospheric oxygen is normally at 21%, which is between 10 to 18% higher than that found in the liver. In the human liver, oxygen concentrations range from 3 to 11%, so in order to represent this, we used a sealed hypoxic incubator to grow the cells at physiological oxygen concentrations, in this case 5% O2. So once established, the models were then exposed to multiple nanomaterials and specific biochemical endpoints analysed to assess the nanomaterial hazards. Firstly, we looked at prolonging the E&M exposure to replicate that of natural human exposure. Then we went on to break the exposure down into smaller repeated doses, as that again was a more realistic way in which humans are exposed to nanomaterials, the small doses repeatedly over extended periods of time. It was also considered that the nanomaterials reaching the liver may have undergone biotransformation from contact with various biological fluids whilst translocating through the body prior to reaching the target organ. So this biotransformation can transform the properties of these materials, and so a tiered testing strategy and SOP for mimicking nanomaterial biotransformation in the liver were developed. To conclude, within this project, we have developed an advanced 3D in vitro Hep-G2 liver model, which is able to support a range of toxicological endpoints for nanomaterial screening. This model has also proven to be successfully transferable amongst both academic and industrial laboratories alike, and alongside this, a more physiologically relevant model and nanomaterial dosing regimes have also been developed as a step closer to a suitable animal testing replacement. If you'd like to find out anything more about the project, the links to our website and social media platforms are listed here. Otherwise, thank you for listening, and I hope you found that interesting.